Assalamu alaikum dear students. Welcome to all of you in our new lecture, that is lecture 4. Dear students, we are discussing chapter 1 whose title is cell structure and function. My dear students, in previous 3 lectures we explained different techniques which are used in the cellular biology. Today, in the continuation of the previous lecture, again we are going to share information regarding some more techniques which are used in cell biology. So let's start. Today we have spectrophotometry. This is a technique which is used to determine the absorption spectra of a solution. What does it mean by absorption spectra of the solution? Every solution, if we pass the light from any solution, it will absorb a particular wavelength, a particular color it will absorb more, and some color it will not be absorbed by the solution. So on the basis of the absorption spectra, we usually determine that what sort of wavelengths are effective in the concerned solution or chemical compound which is in solution. Now, the instrument which is used to perform spectrometry is called spectrophotometer. Now, where this technique is applied? My dear students, it is applied usually for the photosynthetic pigment. What are photosynthetic pigments? Photosynthetic pigments are those pigments which perform the function of the photosynthesis. All of you are very well aware about the chlorophyll, carotenoids. Chlorophyll and carotenoids are the pigments which perform the process of photosynthesis. So, my dear students, usually through this technique we find out that which wavelength is effective in the photosynthetic pigment. Along with that, it is also used for the different compounds to determine what sort of wavelength it is absorbing and what sort of wavelength it is useless for that compound. Now come to the mechanism. This is a light source and here we have lens, and the lens, from the lens, the light comes to the prism. All of you know that light is split into a spectrum. Okay, I just use here red, green, blue and black. It doesn't mean that black is a part of the spectrum, but 3 color markers were available, so I just used the black one here, but it's a 7 colors spectrum. All of you are well aware about that. Then a wavelength selector we have which select a specific wavelength in all this spectra, and this is our compound which we need to determine its absorption spectra. Now it is the detector or photocell, and it is the digital display which will tell us the wavelength, that what sort of wavelength was effective. Another example, if I want to share with you, I got 2 solutions, that is saccharose, simple saccharose solutions, and these saccharose solutions are usually having 2 concentrations. One is of greater concentration, and in one solution the concentration of the saccharose or table sugar is very less. When I pass the light, I will call it I0 or I0, and this is the light which comes out from the solution. This is IK. Now this is another solution which have low concentration of the saccharose solution. If I pass the light from this solution, so what will happen? The light which comes out is denoted by I2. Now the I2 amount will be less than the I1 amount is less than I2. Why? Because there are more particles which are present, which are colliding with the photons and which doesn't allow the light to pass. And here we have low concentration of the sucrose or low concentration of the sugar. That's why the light, more amount of light is permitted, is allowed to pass through. That's why the I2 amount will be high as compared to the IA amount. The cell case is present in our rivers that when we have more turbidity. Turbidity means when we have more soil particles in the water, there is no visibility. We can't see anything what is present in the river. This is called turbidity. Turbidity means you have more soil particles dissolved in the water. So how much from here it is proved that how much we increase the concentration of the solution, that much its absorption will be increased and the light passing phenomena will decrease. Okay, and another in the example, photosynthetic pigment like chlorophyll A, B absorb more red and blue color, and they are thought to be more effective in the photosynthetic process. So it is called the absorption spectra, along with this is called as the action spectra, because these are the two main colors which are absorbed in large amount. If we come to the graph, in this graph, this is the wavelength and this is the absorption spectra. My dear students, if you see 400 nanometer, which is the wavelength of blue color, the chlorophyll A is showing its high amount, the graph is high, and next this graph is high here in the 700 nanometer. But if you see chlorophyll B also similar to chlorophyll A. But keratinide, if we observe keratinides, the keratinides usually we see that it is absorbing light, more light in 600, between 500 and 600 nanometer, and in the absorption spectra of the chlorophyll A and chlorophyll B is not effective in the keratinides. Keratinides are also photosynthetic pigment. It is important to remind you, just like chlorophyll, keratinides are also photosynthetic pigment and they play a vital role in the process of photosynthesis. So my dear students, it was all about the spectrophotometry, and now next we are going to discuss microscopy and micrometry. After that, we will start the cellular organelles. Okay, dear students, next we have microscopy. First of all, we have to define the microscopy, the study of objects by using microscope. Now, what is microscope? It is the instrument which is used to see the objects which can't be seen with naked eye. My dear students, there are, even in cellular biology, if I am talking about the cellular biology, the cells of plants and animals are not visible, majority of the cells are not visible under the naked eye. It needs an instrument for the study, and the microscope is a study through which we study those objects which can't be seen with naked eye. So in the cellular biology especially, this technique is very important, and without microscopy or without microscope, we cannot study the cellular structure especially. Now, what are the types of the microscope? One is called light microscope and one is called electron microscope. Now, the light microscope is the source of radiation is light here, and it have two types usually: simple microscope, light microscope, and compound microscope. The simple microscope usually contains single lens, and the compound microscope have more lenses. You can change, if you want to magnify an object more, you will change the lenses. It usually have two lenses. One is called as eyepiece or ocular lens, and second one is called nose piece or objective lens. These two lenses are mainly used in the compound microscope. Now, the resolution power of the compound microscope is the 250 nanometer or 0.25 micrometer is the resolution. What is the resolution? I am coming on the resolution. Magnification is 4000x. What is magnification? It is also coming. I will explain it. The electron microscope is its name indicates electron. The source of radiation is the beam of electron, and it is thought to be the most powerful microscope because it can magnify an object up to 1 million times. It can magnify an object, and its resolution power is also very high. So it has two types: scanning electron microscope and transmission electron microscope, or TEM, which is called TEM. Now, scanning electron microscope usually tells us about the surface study in 3D structure, and we can take the images which are called electron micrographs through SIM. Now, what is resolution? The resolution is the capacity of the lens to differentiate between two adjacent points. These are the two adjacent points, for example. Resolution is the ability that how you differentiate between the two adjacent points. Now, the human eye can differentiate the 2 points which are away from each other by the distance of 0.1 millimeter. But the microscopes can magnify an object up to resolution is 0.25 micrometer. It will tell us about the difference between 2 points which are very close. Now, magnification. Magnification is the capacity of lens to enlarge an object from its original size. How much it enlarge an object from its original size, that is called magnification. It is expressed by x. If we say 10x, so 10x means 10 times it will enlarge. Like I told you, the magnification power of the compound microscope is 4000x, means 4000 times it can enlarge an object from its original size. So, my dear students, it was all about the microscopy and the microscope. Now, we are leading towards the final technique, that is called micrometry. Now, let's discuss the micrometry. Okay, dear students, the next is micrometry. This is the technique which is used to measure an object under microscope. My dear students, this is a technique by which we can measure any object which we are studying under the microscope. It has two types: ocular micrometer and stage micrometer. The ocular micrometer are the eyepiece micrometer. The eyepiece is the first lens that is present at the start of the microscope, where we see the objects, and which is near, we keep that lens, our eye near to that lens, that is called eyepiece. In the eyepiece lens, usually we can superimpose a micrometer, and it will tell us that what is the size of the object which we are studying. Second is the stage micrometer. Stage is the part of the microscope where we keep the slide, and slide is a glass on which specimen is located. So there is a micrometer also present here, which can tell us the size of the object. Now this is ocular micrometer. The ocular micrometer, if you see, this is a disc of plastic or glass, and this is the scale. If we enlarge the scale, it tells us about the size of the object, that how much the object is enlarged. My dear students, it was all about the technique, different techniques which are used in the cellular biology. And now we will start the structure of the cell. So let's start the structure of the cell. So let me clear this first. Cell. The basic unit of structure and function of all living organisms. In the start we discussed cell theory. Now what it have two types of cell structures we will explain. One is plant cell, and second one is animal cell. Both of the cells are different from one another in some particular structures. Like plant cell have cell wall, and the first structure of the cell, which we will explain inshallah in our coming lecture, will be the cell wall. Then the plasma and the animal cell usually start from the plasma membrane. Chloroplast it have, which is not present in the animal cell. And here we have centrioles, which are present in the animal cell but not in plant cell. There are so many other differences also, like vacuole, single large vacuole is present. And here we have many small vacuoles. Both of the structures are quite different from one another in few aspects only. So in our coming lecture, inshallah, we will start the explanation of different organelles, structures that are present in the cells of plants and animals. Hope that you got good understanding of the today's lecture. Hope if still you have any problem, you can ask your question, you can place your question in the Zoom online session. Stay in home, stay blessed. Allah Hafiz.